Hello everyone, welcome to the new session of Biochemistry of Lipids. So in this session, we are going to study about lipids, right? So in lipids, we are going to see about the characteristic of lipids, right? General characteristic of lipids. Then we will study about the fatty acid, how it is made and what are the different types of fatty acid. Okay. And then we will study about aggregates of fatty acid in aqueous solution, that is called as the micelles. So all these things we are going to study about in this particular session. So, lipids are organic molecules that are soluble in organic solvents such as chloroform, methanol, but sparingly soluble in the aqueous solutions. It means that these are the different organic solvent like chloroform, methanol, right? These are the organic solvents. So the lipid is having the characteristic that they get easily solubilized in these organic solutions, or you can say the organic solvent, but they are not very easily soluble in aqueous solution. That is why it is said that but sparingly soluble in aqueous solutions. These solubility properties arise since the lipids are mostly hydrophobic in nature. The solubility of this lipid in case of organic solvent is due to the hydrophobic nature of this lipid. And one type triglyceride is used for the energy storage since they are highly reduced and get oxidized to release the energy. So this triglycerides, fatty acids, they are having lots and lots of energy. So when the extra lipid is there in the body, that lipid is stored in the form of triglyceride, right? That is the energy storage. And they get reduced and get oxidized to release energy as well. Their hydrophobic nature allows them to pack efficiently through self-association in an aqueous environment. So they are hydrophobic in nature. Hydrophobic in nature means they want to get away from the water, right? And so what they do, they themselves pack in a self-association aqueous environment. So if this is the aqueous environment, the fatty acids or the triglyceride will be seen in this case. The lipids can be seen in this form. They get organized themselves in a form of a pack, right? And that is called as micelles. So that we will see in our coming slides. So, triglycerides are also used for insulation since they conduct heat poorly. So, the triglyceride is having one of the capacity that it work as a insulation, right? This is the characteristic, one of the characteristic of the triglyceride, that they are providing insulation to the body. Okay, and they conduct heat poorly. So they do not conduct heat poorly, so the heat is trapped in that, and which is good if you live in a cold climate, but bad if you wish to dissipate heat in hot one. So once the organism that are living in a very cold region, right, so they are having lots and lots of fat deposit under their skin. And why so? Because it provides a sort of insulation. The heat of the body is not passed out in the environment, and in this way they keep themselves warm. But this is not good for the organism if they are having the capacity that they can store this triglyceride, right? And they are living in a hot region. So at that time, the heat of the body will not get lost, and ultimately there is accumulation of the heat because the heat is coming from the outside also, right? So in that way, the body wants to become cool, and the body cannot getting cooler because the body is having the insulation of the this triglycerides or the lipids. So this is not good for the organism that are living in the hotter region. Now the triglyceride also offer padding and mechanical protection from the shocks. So this is a very important organism because the high deposition of this lipid, right, they are working as an insulation and they are working as a padding and mechanical protection from the shock. They are providing the mechanical protections from the shock as well. So this is some of the characteristic. Now another type of lipid forms a membrane bilayer. So these are some of the lipids. We can see the lipids that are deposited below the skin, below the uh, outer layer of the skin, right? But there are some of the lipids that are present in every membrane. Lipids are one of the major component of the uh, plasma membrane or the cell membrane. So how they are present? They are present as a bilayer. So suppose like this, okay, and this is the bilayer. Now this is one of the lipid molecule. It is having 2 point. The one is the tail region, that is hydrophobic tail region, and this head region is hydrophilic hydrophilic head region. So the two thing is there, head and tail. The tail region is the hydrophobic, so that they want to get away from the water, so that are present in the inner side, right? Both the layer, this is first layer, this is the second layer. Now if you see the tail, they are present in the inner side, whereas the head region, that is the hydrophilic, that are present outer side. So this membrane bilayer separates cellular content from the outside environment or separate intercellular. So suppose this is the plasma membrane, and this plasma membrane is made up of what? Lipid bilayer, and also some proteins are there. So suppose this is the protein layer and this is the lipid bilayer. So here you can see this protein is there, right? Protein is there. Now this is the plasma membrane layer. So this is how the plasma membrane look like. We have just draw here, and this plasma membrane is, this is the outer environment of the cell, right? And this is the inner. So here you can see this membrane is dividing the keeping this inner component of the cell away from the outer environment of the cell. So this is one of the very important factor of the cell, that the lipid is component of the plasma membrane, and this is helping the cell to keep it away from the outer environment, right? Or separate intercellular compartment, that is the organelles, from the cytoplasm. So inside the cytoplasm also, like you can see lots and lots of cell organelles, like nucleus is there, chloroplast is there, right? Lysosome is there, mitochondria is there. So every organelle is made up of, again, a membrane, and this membrane is again made up of lipid and protein. The component of the this lipid and protein in the plasma membrane, as if we compare with the membrane of any organelle, you will find the component is not the same. Means 52% is protein and 48% is around lipid in the plasma membrane, but this may vary according to the organelle membrane. Okay. So some lipids are released from the cell to signal other cells to change the specific stimuli process, and called as the cell signaling. So some lipids are released from the cell. So they are, this lipids are also working as a cell signaling process, right? Lipids are working as a cell signaling process. Now some suppose this is a cell, right? And some steroid hormone is coming, so it may easily get passed inside the cell. Hmm? But if the any proteinaceous signal is coming, then it has to get entered through a specific protein channel. But if any steroid hormone is there, then it can get easily enter through the lipid bilayer, because the lipid, this steroid hormone nature is also lipid only, right? So lipids are released from the cell to signal other cells to change the specific stimuli in a process called cell signaling. So they are also taking active part in the cell signaling process. From a more molecular perspective, lipid can act as a cofactor for enzyme, pigments, antioxidants, and water repellents. So they are also the component. These lipids are also the components of a cofactor for enzyme. There are many enzymes. They are having a specific cofactor. Right. So some lipids are working as a cofactor in enzyme. Some lipids are also a part of the pigments. Some lipids are also the parts of the antioxidants and water repellents. So lipids are also very important part of these particular biomolecules. As we saw with proteins, lipid structure mediate their function. So we have studied about the structure of the protein, how specific they are, how they are folding is responsible for their specific structure, surface, right? And then the function as well. So the protein particular structure corresponds to its particular function. In the same way, the lipid structure mediate their function. So how the lipids structure is there, according to that only their function is also mediated. Fatty acid can be free or covalently linked by the ester or amide link to base molecules like triglycerides or membrane lipids. So, if we talk about the question based on the lipids, so this is a very important part because fatty acids or the free covalently linked ester, so they are having what type of bond? They are having the ester bond. They are having the ester bond, right? So they are ester bond or they are having the amide link to the base molecules like triglycerides or membrane lipids. So these fatty acids are attached to the triglycerides or membrane lipid with the help of ester or the amide link. Now, the key principles that we learned with our study of the protein, that structure determines the function, means the function of any particular biomolecules depend upon its structure, and that we have already seen in the case of protein. So the protein structure is having some conformation. We have studied about the protein is having the 4 level of conformation: primary, secondary, tertiary, and quaternary structure, and that the specific structure determine its function as well. Any alteration or any structure that is away from the normal will not perform the particular function. So structure is very, very important, and that is also applied in the case of lipid as well. The figure below shows the 3 different types of molecules of free fatty acid. So here you can see the free fatty acid. This is the head region, this is the tail region, and a wax. So here you can see the wax structure, and here you can see the fatty acid containing the lipid esterified. It means ester bond must be there, and this is a very important point, right? So the fatty acids are attached with the lipids with the ester bond. Each has a different properties leading to the different function. So here we have seen 3 different types of that is the free the fatty acid, right? Waxes and the fatty acid that are attached with the lipid. So each is having this different properties leading to the different functions. Waxes are instance are very non-polar and water insoluble. So basically they are having most of the component as a non-polar, in the case of waxes, and that is why waxes are not soluble in water, they are insoluble, right? So sometimes it may be asked, the waxes are insoluble in water because of polar and water soluble, non-polar and water soluble, uh, polar and water insoluble, non-polar water insoluble. So the fourth option, that is non-polar water insoluble, that is correct, because the waxes, the component of the waxes, they are non-polar as well as they are water insoluble. They are amorphous solid at room temperature. So at the room temperature, they are solid in nature. But depending upon their structure, they can easily melt to form high viscosity liquid. So they easily melted. So they are amorphous solid at the room temperature. But when the temperature is high, they get melted and they become high viscosity liquid. So this is some of the characteristic of the fatty acid. So they are used as a coating on the surface of leaves to prevent the water loss. So again, they are also present on the surface of the leaves. Okay, and that minimize the water loss due to the heat. Now the glycolipids, that is the glyco as a contain a monosaccharide group, are the constituent of the membrane bilayer. Now these glycolipids, okay, they are the constituents of the membrane bilayer. These glycolipid means the lipid molecules are there, right? Like this. Lipid molecules are there. Now, some glucose molecules are attached with it. That is called glycosylation, and that lipid is called as glycolipids. Not only the glucose, that may be any type of carbohydrate that get attached to the lipid bilayer, then it is called as glycolipids. And they are the constituents of the membrane layer, bilayer, right? So if we talk about the plasma membrane, then there is a protein, then there is a lipid, and there are also some carbohydrates. Okay, very less amount of carbohydrate is also present. Fatty acids vary in length. Now, their length varies, usually contain an even number of carbon atoms and can be saturated, means they have no double bond in the acyl chain, means single bonded, like this, single bonded. All this are present in the single bonded. No double bond is there, right? So that is called as saturated hydrocarbons, or unsaturated with either one monounsaturated or multiple polyunsaturated cis double bond is present. Okay, so the fatty acid is having, if they are not having any double bond, then they are called as saturated fatty acid, and if they are having any double bond, then they are called as unsaturated fatty acid. The double bond are not conjugated, as they are separated by the methylene spacer. So they are not conjugated. They are not attached with any type of conjugated and conjugation, as they are separated by the methylene group. Every time you can see they are is having the space of CH2. Then that is called a spacer as well. So fatty acid can be named in many ways. So here we can see the symbolic name. We will have a symbolic name. Then it is given as X and Y. Then this is the delta A, B, C, where X is the number of carbon atom in the chain. So this X will be number of carbon atom in the chain. Carbon atom in the chain, right? Then what does Y represent? Right? Y is the number of double bonds. So this is representing the double bond. Right? And A, B, C are the position of the start of the double bond containing from the C1. So if there are C1, C2, C3 like this, and at the between 3 and 4 they are having the double bond, then it will be represented as 3. Okay, so the double bond counting from C1, the carboxyl carbon. Now the double bonds are usually is cis form. So systematic name using the IUPAC nomenclature. So this is the name that is provided to the fatty acid by the IUPAC, International Union of Pure and Applied Chemistry nomenclature. The systemic name given to the number of carbon atom in the chain, that is hexadecanoic acid. So hexadecanoic means 16, but it is not having any double bond because it is written here. So this is X is to Y and X is here 16. So the number of carbon present in the chain, that is 16. How many double bond is there? No double bond because it is written 0 here, right? So fatty is unsaturated. The base name reflect the number of double bond, that is the octadecanoic acid for 18,1 at delta 9. So what does this show? This is the 18, that is the number of carbon atom, right? And 1 means at first between 1 and 2 there is a presence of double bond. One double bond is there, right? And which position the double bond is there? The double bond is present at the 9th carbon, and the like this the octadectyrenic acid, that is the 18 is to 3 delta 9, 12 and 15 means 3 double bonds are there, right? One as the 9th position, second is the 12th position, and third double bond is at the 15th position. Now what is the common name? The common name is oleic acid, that is 18,1 delta 9, which is found in high concentration in olive acid. So this is the oleic acid, right? This is the oleic acid. Oleic acid is having 18 carbon atom chain, and there is one single bond, one single double bond, and which position? Between 9th and 10th. So it will be written as 9. So here we can see fatty acid nomenclature. So here 1, 2, 3, 4, 5, 6, 7, 8, 11, 12. So 12 carbon we can see here. Okay, 12 carbon chain and no double bond. You can see there is no double bond is there. So 12 is to 0. It is like written like this. Again, this you can see this, the carbon chain is of 16. So 16 is to 0. Now here you can see 1, 2, 3, 4, right? And till 18. So it is having 18 number of carbon chain. The chain is having 18 carbon and one double bond is there. The double bond is present at 9 and 10th position. So here we will write it as delta 9. One double bond, and where it is present? At 9th position. So like this we can write the fatty acid nomenclature. There is an alternative to the symbolic representation fatty acid in which the carbon atom are numbered from the distal end, their N or omega end at the acyl chain, the opposite end from the alpha carbon. So in this way also we can do the naming. Hence 18 is to 3 delta 9, 12, 15 could be written as 18 is to 3 omega 3. Or 18 is to 3 N3. So in this way also you can write it, where the terminal C is numbered 1 and the first double bond start at the 3rd position, right? Because it is having 18, total number of carbon at, in the chain is 18, and the last here, the last double bond is present at the 15th position. But if you are writing in this particular form, then it will be at 3rd position, right? At the distal end, if you start counting the carbon atom from the distal end, then this 15 will be your 3rd carbon, right? So that is why it is written as omega 3. The most common saturated fatty acid found in the biochemistry textbook are listed in the table. Now, in the next slide, we are having a table. So, we will see these are some of the common saturated fatty acid that is very common in the field of biochemistry. Now, how the melting point increases with the length of the hydrocarbon chain, that we are going to see. This arises from the increasing the non-covalent induced dipole attraction between the long chains, right? So the dipole and dipole attraction we can see here. So here are some of the saturated fatty acid. So 12:0 means no double bond is there. Lauric acid, the systemic name is dodecanoic acid and its formula is CH3CH2 whole 10, means this is repeated 10 times, and COOH. The melting point is 44.2. Myristic acid 14:4 tetradecanoic acid and its structure is CH3CH2 whole 12, means CH2 is repeated 12 times and then COOH. Melting point is 52. Palmitic acid. This is very common. Palmitic acid is one of the very common fatty acid that is found in the living cells. 16:0 hexadecanoic acid, CH3 CH2 whole 14 COOH 63.1. 18:1 stearic acid, that is octadecanoic acid, CH3 CH2 whole 16 COOH 69.6. Archidic acid, that is 20:0. Eoxonic acid, that is CH3 CH2 whole 18 COOH, and the melting point is 75.4. Now, this is also some the table showing the common unsaturated fatty acid. Now, here we are we will study about some of the unsaturated fatty acid that is found in the living organisms, right? So, arachidonic acid, that is omega-6 fatty acid, while docosahexaenic acid, that is an omega-3 fatty acid, right? So here we can see they are decreasing the melting point, right? So the common name palmitolic acid, that is 1619. So 16 is the total number of carbon present in the chain, and there is one double bond. What is the position of the double bond? Between 9 and 10 carbon. So the ninth position is having the double bond. So hexadecanoic acid, and it is written as CH3 CH2 whole 5 CH double bond CH CH2 7 COOH. So this is the melting point of this hexadecanoic acid. Another unsaturated fatty acid, oleic acid, that is 18:1 delta 9. Again, the carbon chain is of 18 carbon, made up of 18 carbon, and it is having one double bond. And what is the position of double bond? That is between 9th and 10. Again, so it will represented as delta 9 here. 9-octadecanoic acid, CH3 CH2 whole 7 CH double bond CH CH2 whole 7 COOH, and the melting point is 13.4. Linoleic acid, that is 18:2 9 and 12. So 2 double bond is there, and one at the position 9 and one another is at the 12th position, right? So 9-12 octadecanoic acid. So here you can see this is the position of the double bond. Another alpha linolenic acid, that is 18:3. 3 double bonds are there, at one at the 9th position, another is the 12th position, and another one more is at the 15th position. So 9, 12 and 15 octadecatrienoic acid. So this is the structure. Melting point is -17. And the last one is 20:4. That is the 4 double bonds are there, one at the 5, 8, 11 and 14th position. Arachidonic acid. So, as there are essential amino acid that cannot be synthesized by human, these are the essential amino acid that we take from the dietary component. So, they are also essential fatty acid that must be supplied by the diet, right? So, this is we know about the amino acid. In the same way, we we are having some fatty acid that we get from our dietary component, and there are only two, one each in the N6 and N3 classes. So let us see that what are those N3 and N6 classes. N6 classes: alpha linoleic acid, that is 18 is to 2 N6, or 18 is to 2 and 9 and 12, means the two double bonds are present at the ninth and 12th position, and it's a biosynthetic precursor of arachidonic acid, that is 20 is to 4 and 6, or 20 is to 4 delta 5 8 11 and 14. So four double bonds are there. This is the N6 class. Another is the N3 class, linolenic acid, that is 18 is to 3 N3, or 18 is to 3 delta 9 12 and 15, is a biosynthetic precursor of eicosapentaenoic acid, that is EPA, and 20 is to 5 and 3, or 20 is to 5, means 5 double bonds are there, that is 5 8 11 14 and 17 position. And to a much smaller extent, docosahexaenic acid, that is DHA, that is 22 is to 6 and 3, that is 22 is to 6 delta 4 7 12 13 16 and 19. So these are the two very important N6 classes and N3 classes that we take these type of fatty acid along with our dietary component. Now, aggregates of fatty acid in the aqueous solution, that is also known as micelles. So, structure determine both the properties and function. Though, if you study about the micelles, then you are going to know about the structure of fatty acid as well as the function. So, it should be obvious that a free unsterified fatty acids are very insoluble in water. So, this particular structure of the micelle shows that how the fatty acid or the lipids are insoluble in water. So when added to water, they saturate the solution at a very low concentration, and then phase separated out into aggregates called micelles. So here we can see the formation of micelle. The structure of micelle formed from dodecyl sulfate, a common detergent, with a sulfate instead of a carboxylate head group, and it is shown below in the figure. So in the next slide we are going to see about that. And all the non-polar carbons and hydrogens of the long alkyl chain are buried. So non-polar, they are non-polar, so they are buried and are not exposed to water. Whereas the sulfate head group are solvent, so they are exposed out. So this is the hydrophilic part, and the hydrophobic part is buried inside. So here you can see these are the tail region, right? Hydrophobic, water hating tail, or you can say the hydrophobic, and the hydrophilic, that is water loving head. In this way you can see they get arranged in the form of micelles. Now we can categorize the lipids based on function or structure. So according to the function, if we talk about, then there are the storage lipid and there are the membrane lipid. So triglyceride is a storage lipid, whereas many different lipids like phospholipids are present in the membrane. Now if we talk about the structure, then glycerolipids, which are glycerol as a backbone for the fatty acid attachment, means the glycerol is a part of this structure, that is a glycerolipids. And sphingolipids, which are sphingosine and act as a backbone. Storage lipid, triglycerols, that is also known as TAGs. Triglycerols contain the majority of fatty acid in the species that store fatty acid for the energy. And you will often see that name triglycerides or triglyceride, but this in term used more in the clinical chemistry and in the industry. So this is all about this session. Thank you very much.